All right, today we're dealing with Mendelian inheritance and phenotypic ratios. For an example, we're going to go over this next slide. Let's go off the bottom here. So our parents, we have homozygous dominant and homozygous recessive, and then their parental gametes. When they cross-pollinate, this is what we are going to get is our first generation. Sorry about that. So when these guys cross-pollinate, we're going to get a heterozygous mix. We have our phenotype and our genotypes. In Mendel's monohybrid cross experiment, these were the results. There's only one genotypic and phenotypic outcome. Heterozygous, which is the genotype of each allele. So the heterozygous organisms with green pods are the phenotype. When these heterozygous green pods were self-crossed, these were the results in this slide. So these are our results. So here we have the ratio of the genotype being GG and GG. And this ratio is 1 to 2 to 1. Sorry about that slide, keeps skipping. So that's our genotype. Then our phenotype. The ratio for this is 3 to 1. And that is dealing with this ratio to 1. This is because any organism with a dominant allele, capital G, exhibits a dominant phenotype. Only those organisms with two recessive alleles, homozygous recessive, exhibits the recessive phenotype, being this. Another type of breeding experiment is called the dihybrid cross. So let's switch slides. So with the dihybrid cross, it is a cross that tests two different traits. In this case, it is a green / yellow and round / wrinkled, as you see here. The first generation, F1, are all yellow and round, only one phenotype. When these F1Ps are bred together, the F2 results in genotypic ratios, which are a little crazy, as we see in this generation of F2. The F2 generation phenotypic ratio is 9 to 3, 3 to 1. This is an important ratio to remember and is a good way to test if certain traits follow expected Mendelian patterns of inheritance. If they don't match this very well, then other things can be going on, such as linkage or mutation. Not everything works that easily in genetics. There are some key complexities beyond Mendelian genetics, which we'll cover in this slide. This is known as incomplete dominance. In this case, neither allele is completely dominant over another, as we see here. Our parents pollinating, creating a pink generation 1. So therefore, this offspring is going to be a mix. Another type is codominance. This occurs when two different alleles affect the phenotype. Affect the phenotype is separate, but distinguishable ways, as we see with these cows here. The next one I want to cover is multiple alleles. Many genes have more than one allele causing many different options, as we see here. That's the way things have been able to form. And the next one to cover is epistasis. And this is when there's a phenotypic expression of one gene that alters the expression of a second gene. Basically, one gene can turn another one off. So up here with our labs, our parents, we're going to separate these codes out. For generation 1, F1. We have our BE, both recessive, or both dominant, our B, which is recessive, and our E, which is dominant, our B, which is dominant, and our E, which is recessive, both recessive here, and then same going down this side, similar pattern. So what we're looking for is the gene that is turning off the other gene, the gene that's being silent or canceled. So here, everything's just dominant. If one recessive, however, mostly everything's dominant, dominant, dominant. We'll go back here. And try to find what is canceling or silencing or turning that other gene off. It's not here, not here. We're starting to see a little bit of a color change. Original color, still have our recessives. One recessive. Starting to see more of a color change. Color change. Here, this is where we see our big color change going to our golden labs. So it looks like this dominant gene is turning off this recessive gene. Dominant turning off recessive. Dominant turning off recessive. And this is just both recessive. So this ties up our Mendelian review.